Okay, good day, ladies and gentlemen, and welcome to this second online lecture on drug design and discovery. So what we did in the previous lecture, just to recap, is we discussed drug design, or gave a definition of drug design, and I also took you through the whole drug design and discovery process, giving you some background on how everything is done. Then we also discussed lead compounds and what a lead compound is and why a lead compound is a very important step of identification in drug design and discovery. And I also showed you some examples of serendipitous discovery of lead compounds. So what we will do in this lecture is to have a look at the sources of lead compounds. And it's very important to note that most drugs are not discovered but are designed from a lead compound. And there are mainly 5 sources of lead compounds, namely that you can 5 sources that you can use to find lead compounds, and that is random screening, through non-random screening, drug metabolism studies, clinical observations, and through rational approaches. We will now go through each of these approaches in which you can identify lead compounds, and I'll show you some examples as well on how this has been used in drug design and discovery. So lead finding approaches, the main lead finding approaches that have been used by a big pharmaceutical company in the past 5 years. Now this big pharma company, it's an international company, it's an innovator company, so it develops new chemical entities, new medicine for marketing. And in the past 5 years in their research, these were the main ways in which they identified new compounds. So first of all, the main area where they found the most lead compounds is through high-throughput screening. We'll discuss high-throughput screening just now. Then also you can see here that they've used virtual screening. About 7% of all of their lead compounds were identified through virtual screening. Now virtual screening is where you use computer software actually to identify new drug compounds. Also, sometimes you can find new lead compounds through just doing a literature search. So, for instance, going through all of the all published papers, have a look and see if you can find any information on a specific lead compound for a specific disease where you've identified a target. And then also patent bust. For instance, patent bust is where you try and make a Me Too drug. They call it a Me Too drug. So it is a drug that has already been patented, but then what you do, you take that compound as your lead compound and you do certain structural modifications, just enough structural modifications, so that it can be considered as a new compound and that you can then get a new patent for that lead compound. So that's known as patent bust. And you can see there are other approaches as well where they've identified, or methods that they've used to identify new lead compounds. We will go through each of these in the next few slides. So the first lead compound identification method is known as random screening. It is also known as high-throughput screening. So high-throughput basically means where you can take thousands and thousands of compounds and you can screen it for a specific biological activity without regard for their structures. Now, this is sometimes used in big pharmaceutical companies or big drug design discovery laboratories where they have libraries of compounds, libraries of organic compounds in their laboratory that makes up thousands and thousands of different compounds, sometimes even millions of compounds that they can take and they can use it in an automated assay process where they can then determine the biological activity of these millions and millions of compounds. But what is important here is that this is without regard of their structure. So it can basically be any organic molecule that they can put through this high-throughput screen. So this method is also the method of choice when nothing is known about the receptor or the enzyme target. So for instance, you know that there's a specific enzyme or a specific target involved in the disease, and you can maybe set up a cellular assay where this enzyme or receptor is present, and then you can determine which type of compounds can bind to this receptor or enzyme target. And by which compounds that binds to this receptor or enzyme target gives you an idea then of what the receptor and enzyme actually looks like. So what protein structure in the body actually looks like. So this is a method that we can use to identify new compounds for receptors and enzymes for which we do not have a lot of information for. So we don't actually know what the enzyme or receptor looks like. So there are two main sources of compounds that we use for random screening. So it's synthetic compounds, compounds that you develop in a chemistry laboratory through organic chemistry, and then also natural products, and then also natural products where you can maybe use plants, where you identify new compounds that you can screen. So you can also, from plants, you can isolate numerous different compounds, thousands and thousands of compounds from plants, and you can use all of that if you've built up a library of natural products that you've isolated. You can also then use that in the random screening method. So one example of two drugs that we all know is the antibiotics, streptomycin and tetracycline, was actually identified through random screening approaches. They were actually these two antibiotics were part of a library of thousands and thousands of compounds. They screened them using a random screening assay or a high-throughput assay. And from those thousands of compounds, they then identified streptomycin and tetracycline as actives for antibiotic effect. So this is just an illustration to show you what high-throughput screening actually is. And as I've shown you in 2 slides back, is that it's the most successful lead finding approach specifically for big pharmaceutical companies. For laboratories such as what we have here at UWC, we don't really use high-throughput screening because it is just too expensive to set up a laboratory, an automated laboratory to do high-throughput screening. What smaller laboratories like my laboratory actually use is known as rational drug design and discovery, and we'll get to rational drug design and discovery in a few slides. So high-throughput screening is basically where you can take, in this case, 1.4 million compounds, and you can set up an automated assay. So if you have 1.4 million compounds and you know what the relevant target is, so the target for a specific disease that you've identified for which there's an unmet medical need, so the target can be any protein like a receptor or an enzyme. So you have the target, you have the compounds, now you develop an automated assay. Now the automated assay, you can see here, sometimes it uses these big machinery that contains what is known as different plates, and each of these plates look like this. So each of these plates are known as well plates, and you can see it has these small wells, like 1, 2, 3, 4, 5, 6, 7, 8 wells in that row. Now each of these wells then contain your relevant target, it contains your compound, and it actually contains also a solvent, like for instance a water or a buffer, water that's been buffered to physiological pH. And what happens then in this well plate, the compound can then react with the target, and then maybe you have an indicator in there, a fluorescent indicator or a UV indicator that can be used then to quantify the activity of your compound on this specific target. So what actually happens, each of these well plates gets placed into this machine, and then what happens from there is you have actually this robotics that's been set up that can take each of these plates and place it into an incubator, maybe at 37 degrees Celsius for 1 hour, can take it out of that incubator, and it can place it in UV detectors or fluorescent detectors that can then detect the indicator and then determine the activity of the compound. So in some cases, maybe the compound will have an effect where it quenches the UV absorbance of the indicator, and by quenching the absorbance of the UV indicator, it gives us then a way in which we can then determine if there's any activity of these compounds in the related target. So from that, then we can then calculate the IC50 values. IC50 values is the inhibitory concentration where the compound then inhibits or antagonizes the relevant target by 50%. So it's a 50% decrease in the activity of that specific target. And then you can see that you will have a lot of data. You'll have gigabytes of data for IC50 values. And then it's further automated by using software models that can actually calculate these IC50 values for you, and it can actually also rank it according to the compounds that are the most active and the compounds that are the least active. And from that, you can then identify, let's say, for instance, the top 100 compounds that showed the best IC50 values using this whole automated process. And then from there, you have identified your new lead compounds that you can then take further into the drug design and discovery approach. Maybe to optimize the activity of the compounds that you've identified, or maybe you do further cytotoxic assays and you find that the compounds are toxic. So maybe you can do some structural modifications to reduce the toxicity of these lead compounds that you've identified. So like I said, high throughput screening, it is the most successful lead finding approach, but it's mainly used by big pharmaceutical companies because, as you can see, it uses a lot of automated processes, uses robotics, and it is very expensive to set up. So you need companies who have a lot of money that they can use in their research. Another approach that you can use to identify lead compounds is known as non-random screening, or also known as targeted or focused screening. So this is where you do screening of compounds that are only related to the active compounds. So you would have seen in high throughput screening, we did high throughput screening on millions and millions of compounds without regard of their structure. So in this case, with non-random screening, you already know what the enzyme and receptor protein looks like. You know which type of compounds may act as inhibitors or antagonists or agonists to the specific protein. You will use that structures that are already known, and you can maybe then develop derivatives of those known compounds or identify derivatives of those those compounds that already have activity. And you only screen those compounds that are structurally related to the active compound. So the structures will look similar. Maybe you have certain functional group modifications or maybe some ring substitutions between the different derivatives that come from the active compound. But in non-random screening, you won't have millions and millions of compounds. Here you will probably have hundreds of compounds, maybe thousands of compounds. So it's a much more focused and target screening. And this non-random screening approach we can do at smaller laboratories. It's not just big laboratories, because non-random screening, you don't have to have that many automated processes. You can actually use postgraduate students to do the non-random screening of hundreds or maybe of thousands of compounds. But it's definitely not an approach that you can use for millions and millions of compounds. So it's more targeted or focused. Another way in which you can identify and where new lead compounds have been identified in the past is by drug metabolism studies. So in vivo metabolites of active drugs are isolated and submitted for biological screening. Now, this is routinely done in drug design and discovery, where you identify metabolites of known drugs. And in some cases, it's not the drug that is responsible for the pharmacological activity, but rather the metabolite which is generated in the body. So one example of this is Silindac. So the anti-inflammatory drug Silindac is not the pharmacologically active agent, but its metabolite is responsible for the therapeutic effect. So it's actually this compound that is responsible for the therapeutic effect. And what scientists have done, this isolated metabolite, they've made derivatives of this isolated metabolite to try and improve the anti-inflammatory action of Silindac and even decrease some of the side effects associated with this anti-inflammatory drug. So sometimes it is the metabolites that are the actual active ingredient, and you can use this then as a lead compound to make derivatives of the active metabolite to try and even further improve the pharmacodynamic as well as pharmacokinetic effects of this lead compound. This is another example of a drug metabolism study. So the antihistamine terfenadine was found to cause abnormal heart rhythm in some patients who also were taking certain antifungal medication, which were found to block the enzyme that metabolizes terfenadine. So fexofenadine is the in vivo metabolite of terfenadine, which also possesses anti-inflammatory or antihistaminic properties, but it can be metabolized even in the presence of the antifungal agent. So fexofenadine is therefore safer than terfenadine. So you can see here that this drug, terfenadrine, is no longer used as the drug because of the adverse effects, but what is currently used as the drug is its metabolite, telfast, which is still active without any of the adverse effects associated with the original drug. So again, in this case, this wasn't just a lead compound that was identified, but this was actually an active metabolite that was identified and which they could then further market as telfast. They've also used this lead compound to make derivatives to try and make it even more active as well as safer. So that's another example of drug metabolism studies. So sometimes you can also find the new lead compounds by just looking at clinical observations. So sometimes a drug candidate will exhibit during clinical trials a second pharmacological activity. That is a usable side effect. This compound may then serve as a lead compound for the second activity. So one example of that is the antihistamine dimenhydrinate. Was evaluated as an anti-allergic drug, and it was found that it possesses a secondary pharmacological activity. It is effective in preventing car sickness in a patient. It then became the most widely used drug for the treatment of all forms of motion sicknesses. So you can see here that it's not always the active with what you actually originally tried to get the drug to be indicated for, but sometimes it has a secondary pharmacological activity, like for instance in the case of Dramamine, and it's now currently being used in motion sickness. So that's another way that through clinical observations you can identify new lead compounds. Another example is the antidepressant bupropion. Also possesses a second pharmacological activity. It was found to help patients to stop smoking and is now the first drug marketed as a smoking cessation aid. And we've all heard about Zyban, which is actually quite effective as a smoking cessation aid. So again, in this case, secondary pharmacological activity was identified, and this led to the lead compound, and that lead compound was then developed into Zyban, in this case as a smoking cessation aid. So you can also use clinical observations to identify new lead compounds that can be developed into new drugs. So rational approaches. Now rational approaches is the main approach that is used by smaller laboratories, smaller drug design and discovery companies, and rational approaches follow the following steps. So you need to identify the cause of the disease. So a rational approach. First of all, you know what the disease is, you know what the cause of the disease is, and then also be able to identify the receptors and enzymes which play a role in the diseases identified. So it is determined if the disease can be treated with an agonist or antagonist of the receptor, or in the case of an enzyme, with an inhibitor of the enzyme. So what you do here is you actually know what the structure of the receptor and the enzyme looks like, the protein structure of that. And then you can then use that protein structure to study it and then determine which type of molecules may interact with the receptor and the enzyme. And what you can also then use, for instance, for lead compounds, is to look at endogenous receptor agonists and antagonists, or enzyme substrates or inhibitors that are used as lead compounds. So you can sometimes then look at the enzyme, look at which type of substrates interact with this enzyme, and use that then as your lead compound. Because if you have a substrate that already interacts with the enzyme, you can maybe do some structural modifications on that substrate, so you develop a new drug based on the substrate with certain functional group modifications in order to make it an inhibitor or an agonist or an antagonist for that specific biological target. So that is known as rational drug design and discovery, and it's also the main drug design and discovery approach that I use in my laboratories. So examples of rational approaches, the one is serotonin. Serotonin we know is a neurotransmitter, but serotonin has also shown some anti-inflammatory activity. And what researchers have done, they've identified the indole structure of serotonin, which has anti-inflammatory action, and then they did certain functional group modifications on the indole structure in order to make it more active, as well as to reduce some of the side effects and also improve the pharmacokinetic profile of this indole type of structure. And what the scientists then came up with is indomethacin. Indomethacin, which is now a very effective anti-inflammatory drug, was developed from serotonin, which is the neurotransmitter which had some slight anti-inflammatory activity, and that led to indomethacin, which is now a very effective anti-inflammatory drug. Other rational approaches that have been used is, for instance, the hormone progesterone served as a lead compound for the contraceptive drug norgestrel, or overall. So they again used the basic structure of progesterone and then did certain functional group modifications on that. So this progesterone, so this is synthetic progesterone, which is then developed into norgestrel, which can be used as a oral contraceptive drug that's quite effective. So that's another way in which rational approaches was used, where they looked at the structure of progesterone, made a derivative of progesterone, and what they found is norgestrel, which is a oral contraceptive drug. So lead discovery approaches, just to give you a summary, we know that random screening or high-throughput screening is where you screen every compound you have, and streptomycin and tetracycline were identified in this way. Non-random screening is known as target or focus screening and only screens compounds related to the active compound. You can also use drug metabolism studies to identify new lead compounds, and the metabolites produced then give us an indication of what type of activities we can see with these metabolites, and also give us an indication of what type of molecules to develop further. You can also use clinical observations, where we've seen some examples on antihistamines that can also be used to relieve motion sickness. And another example is even Viagra, that was previously used as an antihypertensive, but it was found then later on to treat erectile dysfunction, and it was developed into a very effective drug that's been used by males worldwide. Also rational approaches, and this rational approaches, the main approach that are used in smaller laboratories is where you identify the biological systems involved in the disease states. You use natural receptor or the ligand or the enzyme substrate as the lead compound, so you get to know what the actual receptor or enzyme looks like, and a known drug can in some cases then also be the lead compound that you know that can interact with this ligand or receptor. And you use that as the lead compound then to further optimize that compound into an active compound that can be taken further into the drug design and discovery process. So guys, that brings me to the end of this second online lecture. I really do hope that you enjoyed it. What you need to do next is please go through lecture 1 again and the second lecture, and please read through my study notes as well, which will then provide you with additional information that is necessary for you to effectively complete this first part of the drug design and discovery lectures. Thank you.